Hi, I'm Dr. Shoshana Schendelman, founder and CEO of Applied Therapeutics. At Applied Therapeutics, we're applying science to transform lives. We're developing drugs for high unmet need indications, fatal or debilitating diseases that have no treatments approved. One of the diseases that we're working on is called SORD deficiency, and that's what I'm here to talk to you about today. What is SORD deficiency? Sorbitol dehydrogenase, or SORD deficiency, is a progressive, debilitating hereditary neuropathy that affects peripheral nerves and motor neurons, and it results in significant disability, loss of sensory function, and decreased mobility. About 3,300, little over 3,000 patients in the United States have SORD deficiency, and about 4,000 patients in Europe. Most patients with SORD deficiency were previously diagnosed as having Charcot-Marie-Tooth type 2 or CMT2 unknown genetic cause, or distal hereditary motor neuropathy or DHMN. Recently, research has shown us that loss of the SORD enzyme or genetic mutations in the SORD gene are responsible for this new disease called SORD deficiency. So there's now an opportunity for patients who previously had kind of a vague disease diagnosis like CMT2 unknown or DHMN to get a specific diagnosis of SORD deficiency. And this research was so important because the SORD enzyme is actually very well studied and well understood. So once we knew the genetic cause behind this type of disease, we immediately understood why patients are having the symptoms that they have and what we call the pathogenic cause, or what's biologically causing this disease to happen. And that was really a breakthrough because it also allowed us to develop potential treatments for SORD deficiency. Patients with SORD deficiency have high sorbitol levels in their blood and tissues. And to understand why, it's important for us to look at the biochemical pathway responsible for metabolism of sorbitol. Sorbitol is what we call a sugar alcohol. So sugar is converted to a sugar alcohol. Glucose is converted to sorbitol by the enzyme aldose reductase. And then in healthy individuals, the next enzyme that follows and metabolizes sorbitol to fructose for energy is sorbitol dehydrogenase. So in healthy individuals, a small percent of our glucose is actually metabolized this way, or a small percent of the sugar in our body. It's not the way that the majority of the sugar in our body is metabolized. About 1% goes through this pathway. And in healthy individuals, sugar or glucose is metabolized to sorbitol, then sorbitol is very quickly metabolized to fructose for energy by the SORD enzyme. So you don't have very high sorbitol levels in your blood if you have both of these enzymes. However, patients with SORD deficiency are missing the SORD enzyme, and so they're unable to metabolize sorbitol. And what happens is that even though a very small percent of our body's glucose goes through this pathway, aldose reductase is converting that small amount of glucose to sorbitol, and it has nowhere to go. There's no way for it to be further broken down or metabolized. And for that reason, sorbitol levels build up to be very high in SORD deficient patients in blood and tissues. In fact, about 100 times higher than the sorbitol levels in the blood of healthy individuals. So what are the symptoms of SORD deficiency? 100% of SORD deficiency patients have lower limb weakness or motor dysfunction. This often presents as difficulty walking, running, climbing stairs. It also results in difficulties with balance and ataxia or gait problems. A little over half of patients with SORD deficiency also have upper limb weakness or coordination issues in their hands and arms. Patients also have tremor, neuropathic pain, and sensory dysfunction. So how do you know if you have SORD deficiency? Most patients with SORD deficiency have previously received a symptomatic diagnosis, even though we didn't know the biochemical cause of their disease, and currently have a diagnosis of either CMT2 unknown, or in other words, CMT2 where we don't know the genetic cause of disease, or DHMN. It's also important to know that SORD deficiency is what we call an autosomal recessive disease. So that means that most patients with SORD deficiency do not have a parent who's affected with the disease. If you do have a parent who's also affected with the same form of neuropathy or motor neuron disease that you have, it's likely that it's a dominant form of disease. So it's probably a different type, not SORD deficiency. And now we can actually test for SORD deficiency via a simple blood test that looks at sorbitol levels in your blood, or via genetic testing. So why is it important to find out if you have SORD deficiency? It's really important, especially in rare diseases like SORD deficiency, for the medical community to better understand the disease and the specific symptoms and issues that are meaningful to SORD deficiency patients. And this allows us to improve diagnosis and also to design better clinical trials. There is an investigational drug being studied in patients with SORD deficiency, and we'll talk about that a little later. But it's important to know if you have SORD deficiency because you may qualify to participate in a clinical trial for one of these investigational drugs. For that reason, Applied Therapeutics is running a free SORD deficiency screening program. So patients can actually screen to find out if they have SORD deficiency via a simple blood test, which can be performed either in your doctor's office or via a home health nursing visit. We're performing this testing in patients aged 16 to 65. And if you're interested in having this free testing service performed, please email us at sword@appliedtherapeutics.com. So now we'll talk a little bit about AT007 and the investigational program or clinical trial program in SORD deficiency. So what is AT007? AT007 is an investigational drug. That means it's permitted by the FDA for use in clinical trials, but it's not yet approved for treatment in SORD deficiency. AT007 is an aldose reductase inhibitor. So it's a drug that blocks the enzyme that comes before sorbitol dehydrogenase. It's the enzyme that's responsible for conversion of glucose to sorbitol. So our goal is by blocking the enzyme aldose reductase, we can actually stop production of sorbitol. And so even though patients are missing the enzyme that metabolizes sorbitol, it shouldn't matter that much because we're stopping production of sorbitol. AT007 has been previously studied in adult healthy volunteers, as well as adult patients with a different rare disease called galactosemia, and pediatric galactosemia patients aged 2 to 17. In clinical trials, AT007 is dosed once daily in a strawberry flavored oral suspension or liquid. So here you can see the mechanism of action of the drug AT007. Again, glucose is normally converted to sorbitol by the enzyme aldose reductase. And if we're able to block that enzyme with AT007, we can stop sorbitol from being formed. And then even though patients with SORD deficiency are missing the next enzyme that's responsible for sorbitol metabolism, it shouldn't matter very much because we're stopping sorbitol from being formed in the first place. We've done a lot of work looking at AT007 in what we call preclinical models, and that includes things like cells from patients with SORD deficiency and animal models of the disease. So AT007 treatment significantly reduced sorbitol levels in SORD deficiency patient fibroblasts. Fibroblasts are cells that are taken from SORD deficiency patients and cultured in petri dishes in the lab. So you can see on this graph that patients with SORD deficiency, even their cells cultured in the lab produce very high levels of sorbitol. And that's the gray bar in the center of the graph. you can see that the sorbitol levels are much higher than people without SORD deficiency or healthy individuals. Cells from healthy individuals are shown in that left-hand blue bar. Now, when we treat the cells from SORD deficiency patients with AT007 in the culture dishes, we're able to really significantly lower sorbitol levels. And that's what the green bar is on the right-hand side. We also studied AT007 in a Drosophila or fruit fly model of SORD deficiency. So in this model, the sorbitol dehydrogenase gene was knocked out of fruit flies. And so you can see that these fruit flies actually have neuronal degeneration as a result of SORD deficiency. And they interestingly had a very similar phenotype or symptoms of disease to actual patients with SORD deficiency. And in fruit flies, that presents as difficulty walking and climbing and similar mobility and motility issues to humans with SORD deficiency. What's great about Drosophila is that we can actually visualize or see their neurons very well. And that's what these beautiful fluorescent pictures are on the left-hand side of the screen. So what we're looking at in the left-hand panel is normal fruit flies. They're called yellow-white fruit flies. And you can see that their neurons are this beautiful, bright fluorescent pattern, and it's sort of uniformly across the page, across this block. And the middle panel is the neurons of fruit flies with SORD deficiency, and their neurons start to degenerate. There's damage to their neurons. And the way that we see this under the microscope is in the form of vacuolar structures, or literally black holes in the neurons. That's what these arrows are pointing to here. So you can see that the neurons really are degenerating in fruit flies with SORD deficiency. And when we treat the SORD deficiency fruit flies with AT007, you no longer see those black holes or vacuolar structures. And you see the nice normal fluorescent pattern that's also there on the left-hand panel in normal fruit flies. And this can actually be quantified. That's what we're seeing on the right-hand graph here, that fruit flies with SORD deficiency have a high number of vacuolar structures or neuronal degeneration. And when we treat them with AT007, then we prevent that neuronal degeneration. And the AT007 treated Drosophila look just like the wild type or normal Drosophila. And also, it's not shown here on the screen, but AT007 treatment also did prevent the mobility and motility and climbing problems that those Drosophila showed as part of the phenotype. So this neuronal degeneration we know is what causes difficulty walking and climbing in fruit flies, just like in patients, and treatment with AT007 prevented that from happening. So that was the data building up to our first clinical trial in SORD deficiency. It's a pilot study, which means it's a very small clinical trial, just to look at what we call proof of concept. So we had data already in fibroblasts from SORD deficiency patients, and then Drosophila with SORD deficiency. And now we wanted to see if AT007 treatment could also prevent sorbitol formation in SORD deficiency patients. So we treated 8 patients with SORD deficiency over about 1 month of treatment. And you can see here on the slide that AT007 treatment very substantially reduced sorbitol levels in patients with SORD deficiency. So all of these patients had very high sorbitol levels in their blood before they were treated with AT007. And that's what we would expect, about 100 times higher than normal sorbitol levels. So the gray bars on this left-hand graph are the starting or baseline sorbitol levels in the blood for each of these 8 patients with SORD deficiency. And the green bars is their level of sorbitol in their blood after treatment with AT007 for about 1 month. So the mean reduction or average reduction across the 8 patients was 66%. And the range, or the high and low values, the lowest percent reduction of sorbitol that we saw was 54%, and the highest sorbitol reduction that we saw was 75%. So that's a really substantial level of sorbitol reduction in the blood, and this was in just 1 month of treatment. So all of this information has come together to help us design a registrational trial, or a clinical trial that should hopefully support approval of AT-007 for SORD deficiency if the results are positive and it continues to be safe and well tolerated. There will be more details on this trial coming soon, but a little bit of a preview. This will be approximately 50 patients with SORD deficiency, and it will be what we call multi-center, so multiple sites in the US and EU. And we'll look at sorbitol reduction, or this biomarker outcome, as well as long-term clinical outcomes, or how patients feel and function over time. So what should you do if you think you have SORD deficiency? Please get tested. It's important to know if you have SORD deficiency. It's important for the medical community to better understand this disease. And it's important that you share your experience. Make sure that if you find out that you have SORD deficiency, that your physician, the medical community, better understands the symptoms of the disease and aspects of SORD deficiency that are important to you in everyday life. We know that in rare diseases, the patient perspective is critically important. Please consider connecting with other SORD deficiency patients. It is a rare disease, but there are over 3,000 patients in the US. And as in all rare diseases, the community is stronger together. And if you qualify, please consider participating in a clinical trial for SORD deficiency. That's the only way that we're going to really be able to bring treatments forward for SORD deficiency. So with that, I really thank you for joining us today. If you'd like to learn more about SORD deficiency, or if you're interested in getting tested, or if you would like more information about our clinical trial program, please email us at sword@appliedtherapeutics.com. Thank you.